All right, so starting out with the stationary phase, this is where we left off, right? So I was just speaking about the fact that the in the stationary phase is usually the thing that doesn't move, it's stationary, right? It's usually the media through which the the different types of separations, right? The different substances will be moving through, right? And we're speaking about the fact that the molecular composition is important, right? So for the silica gel, right? Within silica gel itself, silica gel is silicon oxide, right? Silicon dioxide, and it has that type of bonding, right? But on the surface, it has OH bonds, right? So that is polar. So that will actually cause interactions with polar species, right? And not so much interactions with non-polar species. And that's the same thing for alumina. It has alumina hydroxide on top, right? So what's happening here is that we're gonna have, let's say, something very polar like benzyl alcohol that will move much slower, right? Benzaldehyde is going to actually be polar, but it's gonna move actually a little bit faster than benzyl alcohol because it's not as polar. And something like ethyl benzene will move swiftly throughout silica gel, right? Because it's not attracted to the stationary phase at all. So we actually have substances will have longer retention times if they're more attracted to the stationary phase because they will take a longer time to actually move through. Make sense? Everything clear now? Hopefully, because you guys should be able to hear me clearly. All right. So that would be that for the silica gel, right? The mobile phase now, right? A fluid which percolates through or along the stationary bed, right? In a defined direction. That is what a mobile phase is, right? It may be a liquid, right? In liquid chromatography, it may be a gas in gas chromatography, or it may be a supercritical fluid in supercritical fluid chromatography, right? So just giving examples of what it can be. In gas chromatography, the expression carrier gas may be used, right, as the mobile phase. The expression carrier gas is used as the mobile phase. And in elution chromatography, the expression eluent is used as the mobile phase, right? So we can call it a mobile phase, but if we're talking about gas chromatography, we tend to call it the carrier gas. That is the same thing as mobile phase. And in elution chromatography, we call it an eluate. Eluent. The mobile phase is called an eluent right there, right? Okay. So this is a checkpoint where I'll stop the first section, right? The first part. Now, this now, right, is just some questions generally, right? So if you can do these questions, right, you should be able to really understand, or you should have a good understanding of what came before. All right. So these questions should be able to guide you to understand the content of what came before. Let me go back. So you must be able to explain the principles upon which chromatography is based. Should be able to do that. Number two, explain the following terms: retention factor, retention time, solvent front, and visualizing agent. So hopefully we can explain those. We must be able to distinguish between the stationary and the mobile phase in chromatography. And number four is an entire question, right? It's an entire statement in which you must be able to calculate the retention factor of the blue pigment in each sample provided in that statement and be able to figure out whether or not there are differences between the two bands, right, of purple coloring. Go ahead. Somebody raise your hand, I think.
Sir,
Go ahead.
for gas liquid chromatography, would some inert gases be like hydrogen, nitrogen, and helium?
Nitrogen as an inert gas. Ooh. I would accept hydrogen and helium, right? Nitrogen, I think it would depend on what you're doing, right? But the inert gases tend to be our noble gases and stuff simple like hydrogen. Alright, but I do assume, yeah, nitrogen could be used under certain circumstances. So those would be some of our carrier gases, right? Our mobile phase. It can be used. Alright. So the types of chromatography, no, the paper chromatography, we know paper chromatography. There's not all the information that I have on this. It should be fine, right? So The stationary phase is just paper, it's paper chromatography. The mobile phase is a liquid, commonly water, but could be ethanol or a non-polar solvent, right? And this is generally how we set it up. Should be fine. So it can be done with drugs, inks, dyes, etc. Right? And we have that there. The mobile phase is usually something like methanol, hexene, or carbon tetrachloride, and the stationary phase is usually a paper or chromatographic plate. All right. So here we see the liquid level that is beneath the starting line, or what we call the baseline, and we have the finish line, or what we call the solvent front. And it's in a beaker covered, and you have the mobile phase here. In this case, the mobile phase is a solution of ammonia, butanol, and water. Right, so you can see those things now. Right, and you can also have it like this in a test tube. This is what we usually did for our chlorophyll thing, right? So chlorophyll A, chlorophyll B, carotene, right? Xanthophyll, right? You can get these different separations, right? Using paper chromatography. You also have thin layer chromatography. Is this xylol? This is xylol. All right. Okay, so let's have a look at this. So chromatography is carried out on a layer of absorbents, right? A layer of absorbent spread on a support backing, a glass plate, right? So you have an absorbent, and then you're going to put that on a glass backing, and that's what you're gonna have a TLC plate, right? As the solvent, the mobile phase, moves up the plate, the components of the mixture partitions itself between the mobile phase and the stationary phase, and the more polar component will spend more time, right, adsorb onto the stationary plate, while the less polar component will actually spend less time dissolve, and it would be dissolved in the mobile phase. All right. So another term that I want you guys to define is adsorption. Right. So I want you guys to define that. As the result, the mixture will be separated into its two components. So in this case, if we separate screen methyl orange, we're gonna have the methyl orange, and we're gonna have a xylol substituent, right? So we can separate stuff like that on a TLC plate, right? This is not specific for you to know, but it's just an example that you can separate methyl orange into these two substituents, right? That's TLC, right? Another run of TLC. So this is the form of adsorption chromatography. Silica, alumina, or cellulose, so stationary phase, are made into a paste and spread on a thin layer of glass or plastic plate. So that's the adsorbent that we're talking about, right? And we generally know how it works, right? So it should be fine. Don't have to go by that over and over again. So these are examples of how the stationary plates, the TLC plates would look. All right, should be fine. Okay. For column chromatography now, the principle is the same as the TLC, right? But it's set up in a different way. So the mobile phase moves over the stationary phase, carrying the components of the mixture along with it, right? The less polar component will spend more time the mobile phase, in the mobile phase, right? And therefore move through the column at a faster rate, right? The less polar one will be the mobile phase, fast going through a faster rate. The more polar one will actually be more absorbed onto the stationary phase and spend a longer time, right? The time a component spends in the column is our retention time, right? So in this diagram now, which one of the substances would have the highest retention time or the longest retention time? That's my question to you guys. Which one of these would have the longest retention time? Blue, red, or green? Hopefully I'm not lagging again. You guys can hear me? not sure if it's just that people are not answering, right? So the blue one would have the longest retention time, the green one would have the least retention time. For column chromatography, no, right? Usually a relatively non-polar solvent is used during the column chromatography. This may be acceptable to remove the less polar component from the column. However, more polar components might have very high retention times with the solvent, and this might require changing the solvent in order to elute these components. So sometimes it will be very, very high with a specific solvent, so we need to change in order to actually move it out. Right? For gas liquid chromatography, GLC, no, right? This is a form of partition chromatography, no. So it's different from adsorption chromatography. No, it's partition chromatography. The stationary phase, right, is a high boiling liquid, right? High boiling point liquid. A long chain hydrocarbon oil supported on a porous inert solid such as silica or alumina. So the mobile phase is a carrier gas like nitrogen, helium, or argon, right? And this is the carrier gas that we see, right? How it works, the mixture to be separated is injected into the instrument via the injection port. As the mixture is carried through the instrument by the gas, these substances that are more soluble in the oil will travel more slowly, and those that than those travel more slowly than those are less soluble, right? So that's what's happening there. So we had an adsorbent solid, right? Are we gonna be having we're having mobile phase as well? Are we gonna be partitioning between the adsorbent solid, right, and the mobile phase? It's a partitioning thing, right? So a schematic diagram of the gas chromatography instrument. So this is generally how it looks and how it works, right? So you push the gas through and stationary phase, and they are separated like that. Okay, and this over here on your right is an example of how it looks attached to a mass spectrometer in order to detect exactly which gas is coming out at what time. All right. So, so here now, right, the glass liquid chromatography now, the column consists of a long coiled tube with an internal diameter of around 4 millimeters, is located inside a thermostatic controlled oven. The oven is either set at a predetermined temperature or gradually increased during the separation, and the column is packed with finely ground di diatom diam diatomaceous, right? Diatomaceous earth, right? I always just call it diatomaceous. I don't know why, but that's the earth that's really there. It's basically a very porous rock, right? This is coated with a high boiling liquid, right? Typically a waxy polymer, and the components of the mixture can either condense on the stationary phase, dissolve in the liquid on the stationary phase, or remain in the gas phase, right? So the rate of separation depends on the boiling point of the compound. Higher boiling points gives us longer retention time. Solubility in the liquid phase, higher solubility equals higher retention time. And the column temperature, high column temperature gives us shorter retention time actually. Right? And here are schematic diagrams showing us what gas chromatography results would look like. Alright, this would be with the mass spec, and this would be generally, right? All right, so that is generally the end of chromatography. What we have left is just applications of chromatographic methods, which we can see here. So paper chromatography is usually used to analyze mixtures of dyes and ink, coloring in food additives, and amino acids in protein hydrolysis, right? Visualizing agent for amino acids tend to be ninhydrin, right? For thin layer chromatography, we tend to commonly use to separate amino acids in blood samples and for analysis of food dyes. Column chromatography is usually used in the biochemical research and hospitals to identify amino acids, peptides, and nucleotides. High performance liquid chromatography we tend to use that in the industry and in hospitals to identify suspected stimulants, doping, and drugs that may be present in athletes or racehorses. So it's really used in that type of industry, right? The sports industry as well. And gas liquid chromatography is very sensitive method, so it's using testing, right? To monitor air and water pollution, to detect identity traces of pesticides or agricultural chemicals in foodstuff, right? And to check dosage of drugs in blood and urine samples. So that's really all we have there for chromatography.